Lab 6, Passive Transport by Soham, Nick, and Neil. Organization. We plan to organize our presentation into 4 videos. Our first video is the introduction to membranes. Our second video is the osmosis experiment. Our third video is the diffusion experiment. And our fourth video is the microscopic sheep blood experiment. Introduction. What are membranes? Membranes are basically one of the most important parts of the cell. They act as a selectively permeable membrane or barrier, and by selectively permeable it means that not everything, and by everything we mean ions, gases, liquids, macromolecules, not everything can easily pass through. By that, you need to either have a protein gradient or like an active transport site. The phospholipid bilayer separates the intracellular environment from the extracellular because they're not the same. The phospholipid bilayer regulates the flow of ions, gases, and macromolecules, which is basically controlling what goes in and out of the cell. And the membrane has a special property known as polarity, and polarity regards the membrane from many molecules or ions that cannot pass through directly, because in the bilayer there are parts known as the fatty acid tails, and those are nonpolar, which basically make the middle layer of the phospholipid bilayer. In this picture, we can clearly see the phospholipids right here, and let's use my pen. Oops, let's make it, yeah, red. Here you can clearly see the phospholipid head. That is the polar part, and the tails are the nonpolar part right here. And in this picture, you can see the phospholipid bilayer, which is right over here. And as previously stated, the nonpolar tails are in the center, while the polar heads are on the ends. And what also regulates the transport of ions, molecules, and gases are proteins, as stated in these structures right here. They regulate the flow and, well, that's true. Yeah, they regulate the flow through active sites, which are when ATP binds as an energy carrier from low concentration, which is extracellular environment, to the high, what's it called? Oh, the high concentration gradient, which is the intracellular or cytosol. And what helps in recognition of molecules are these carbohydrates, which are here. And they help in cell recognition, as in what molecules are going to enter the cell or what cells are like close together. And these special molecules, which are known as cholesterol, we will talk about later. Here, phospholipids are the most abundant part of the bilayer. They are made of the polar head and the nonpolar tails. And by this, the polar head means that it is hydrophilic, which means it is water-liking or loves water. And the nonpolar tails mean hydrophobic, which are afraid of water. And the point we're trying to make is that the polar heads basically protect the nonpolar tails, because the nonpolar tails wouldn't be able to dissolve. Well, they would be converted into smaller parts if they weren't protected by the polar heads. Proteins. Proteins function as transporters, which can be further classified as channel proteins, pumps, or symporters. These all, well, not all of them, but pumps and channel proteins require energy most of the time, and symporters, some of them do and some of them don't. Receptors, they need a hormone which is released by the pituitary gland. Well, we don't need to go into that, but they are binding sites for the protein that allow the gradient channel to open and allow molecules to pass through. And as I said before, the glycoprotein, which is used for the cell recognition, that is a intermembrane protein with a carbohydrate attached to it. Cholesterol. Cholesterol is a steroid, a different classification of a lipid. They are found at the nonpolar tails, well, not at, but in the area of the nonpolar tails because they're hydrophobic. And the function of this cholesterol is to maintain the membrane fluidity. Based on the temperature of the environment, cholesterol either keep it fluid or help solidify it from, turning into fluid. Carbohydrates. Carbohydrates are used for cell recognition. In glycosylation, it's the process as when carbohydrates are added to a protein, as in a intermembrane protein, to help with cell recognition. And seen here, carbohydrates can either be attached to the intermembrane protein, which is here, or the lipids, which are here.